we were contacted by a pharmacy out of Connecticut, and they were testing Zantac, which is ranitidine, the active ingredients, and they were testing it by gas chromatography. And gas chromatography, you're heating the sample enormously in order to volatilize certain impurities. And they were observing lots and lots of nitrosamine, specifically N-nitrosodimethylamine, NDMA. Millions of nanograms of nitrosamine, which is just completely unacceptable. NDMA, according to the FDA, you can have only 96 nanograms per day. Our investigation effectively unraveled what's happening with the ranitidine. Effectively, ranitidine was breaking down, and the breakdown product of ranitidine was the NDMA.
What does it take to build a company around a scientific discovery that reshapes public health policy? Ron Najafi has done exactly that. From his early days as a bench chemist to founding NovaBay Pharmaceuticals and taking it public, to leading Emery Pharma, a CRO deeply committed to science, CMC, and bioanalytical testing, Ron's career is a masterclass in turning scientific curiosity into real-world impact. Today, he shares where that journey began. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Ron. It's good to have you on today.
Thank you, David. It's a pleasure to be talking to you again.
Ron, share something that you believe about bioprocess development that most people disagree with.
I think one of the things that people should not disagree with is to do proper homework on CMC development, because that's going to be one of the biggest holdups as the project moves from preclinical to preclinical and so forth and so on. Because understanding CMC, understanding the challenges associated with manufacturing, with analytical chemistry of your drug substance, drug product, biologics, are essential to your success. And also it's essential to your pharmacology, to your toxicology. So you need to know as much as possible about your chemistry, manufacturing, and controls. And I say that with some intentionality. Chemistry is really how you make your product, how you make your compound, how you make your biologics. You really need to know it. And it makes a big difference in how you're going to ultimately manufacture the product. Chemistry, manufacturing, and manufacturability, whether this is going to cost you $1 million to make it, or you'll never, ever be able to make it. So those are all connected, related. And then, of course, controls really refers to quality controls. It refers to how you're going to test the product and how you're going to maintain the product, how you're going to have the shelf life of the product. So those are things that there are agreements and disagreements, and I think there should be some attention made to all of that, the CMC at preclinical and pre-preclinical stage, because FDA cares about it, EMA cares about it, and we need to make sure that we also care about those things, because that could be a major holdup for anybody developing either small molecules or biologics.
Very good reminder. Thank you, Ron. And this is a point I've been stressing multiple times here in the podcast. CMC is important and should not be an afterthought. Ron, you have been on a fascinating journey for several decades. Draw us into your story. What first sparked your passion for science, and what were some of the interesting pit stops along the way?
No, I actually thought about it. One of the things that sparked my passion was when I was in high school, and when I had a high school chemistry professor who was very engaged in chemistry. And when the time came to go to the laboratory and do basically experiments, we were given a lab coat. And I remember vividly how I was carrying my lab coat. I felt that lab coat was like a cape, was like a Superman cape. It gave me authority. It gave me something that I never had before. So that was one spark. The second spark was when I was at University of San Francisco, and I met my professor that later became my major professor, where I got bachelor's and master's. He really encouraged me into doing synthetic chemistry. And I remember we were up till 4 a.m. sometime doing chemistry, making 9-BBN or doing synthetic chemistry. Organoboron chemistry was very exciting. It was Nobel Prize was given to H.C. Brown in 1978, I believe. And it was very exciting time. And the second part was really when he gave me the key to the lab, was a major milestone. I felt like now I have a lab, I have a desk. So those 2 points were 2 sparks in my life. It was like a quantum leap from one place, from one location to the next location. And it was transformative to most people. That maybe not that big of a deal, but to me, getting a lab coat in high school and going to the lab at the University of San Francisco, getting a key to the lab and having my own desk in the lab was very significant for me.
And what were some other interesting stops and experiences you had along your, I believe it's more than 3 decades already now, as an entrepreneur, scientifically? Tell us your story.
I have to warn your listeners that time flies. 3 decades flew by so fast, but I never watched the clock, David. I have never been a clock watcher. I work. I enjoyed it. So I basically, one thing led to another. I got my bachelor's and master's with this professor at USF and ended up going to UC Davis and continuing on actually organoboron chemistry, which was that Nobel Prize winning sort of technology and chemistry that H.C. Brown developed. So I worked for several H.C. Brown professors, and I got my PhD from UC Davis. Shortly after, I was really hoping to get a job in San Francisco Bay Area. My family lived, but unfortunately, there were no jobs. There was just Genentech, which wasn't really doing synthetic chemistry. There was Dow Chemical and Clorox and a few other small, very tiny biotech. And there was nothing in the Bay Area, nothing like it is today. Absolutely nothing. This is 30 years ago, more than 30 years ago. And basically, I applied to Hewlett-Packard in Corvallis, Oregon. I got rejected. And my boss, George Zweifel, basically said, Ron, if you want to go work for Aldrich Chemical Company, I can get an interview for you. So I said, okay, I'll go to Aldrich. It was in Wisconsin. I go like, no, I don't want to go to Wisconsin, but I ended up going there. And I went to Wisconsin and they made me an offer right there and then. I said, okay. I interviewed in July, which was beautiful, warm, sunny. And My job started in February and it was blizzard, snow. So I spent 2.5 years or so at Aldrich Chemical Company. I was involved with organosilicon, organoboron chemistry. And then I got a job offer through a recruiter to go work for Rhompolenk Rohr in Philadelphia. And I actually practiced organoboron chemistry there, working for Rhompolenk Rohr, and was involved with being a research scientist. I made molecules day in, day out. And I was there also 2.5 years. One of the things that I'm very proud of, I actually started a symposium there called Visions in Chemistry, which actually continued up to the pandemic, and to focus on one technology, one type of chemistry. And our very first symposium, which I was the organizer, was all about organoboron chemistry. And I invited H.C. Brown and all the organoboron professors and scientists and my former boss from Aldrich. And then I really wanted to come back to California. So the same recruiter that got me the job at RPR introduced me to two potential positions, one at San Diego and one in San Francisco Bay Area. And I took the San Francisco Bay Area, interviewed. It was Applied Biosystem. And being an instrument company, DNA synthesis, I got involved in synthesis of phosphoramidite chemistry, and I got deep into chemistry, manufacturing, control. There was a problem with phosphoramidite chemistry, and it was a problem that caused problem in the synthesis of DNA. So we have the phosphoramidites for AGTC, and those phosphoramidites were not giving us good quality primers. I dived deep essentially into the analytics of it and figured out what was causing it, removed it from small scale and also large scale, and I was awarded the highest chemistry award at Applied Biosystem amongst 1,200 PhDs. And let everybody know, Applied Biosystem people will know about it. And basically, that same year, I also invented a product called EcoFunnel. And that product, essentially, it's a funnel that screws on top of a bottle. EcoFunnel, ECO funnel. I invented it to prevent pollution from the lab. I've been involved in the safety committees throughout every company I was at. I cared about the environment. I cared about the people. I didn't want people to get exposed to chemicals unnecessarily, and also didn't want things to evaporate into the fume hood. You know, you put a gallon of solvent into the fume hood and leave it open. In a few days, there will be no liquid. There will be no solvents. Everything is evaporated. So I essentially created a trap to prevent evaporation. At the same time, you could dump your liquid waste into the bottle. So I invented that product. Applied Biosystem was very forward-looking and early adopter in everything. And they ordered the product. They said, Ron, go ahead, make it for us. Go ahead and patent it for yourself. I developed it by and large during my breaks and during my lunch hours and evenings and weekends. And they didn't want to have anything to do with the patent. I patented it. Essentially made a company around it. It's called CP Lab Safety. This year, we're celebrating 30 years of CP Lab Safety. So I ran that company for a few years. I made sure the company is successful. It was a challenging product because people used to tell me, why should I buy this product? Right now, I'm evaporating all this waste solvents, hazardous waste, into the fume hood, and I'm not paying anything. But if I buy your product, I have to pay for your product and then also pay for the proper disposal of all these solvents. I said, yeah, that's the right thing to do. And if you evaporate solvents into the fume hood, essentially it's like dumping solvents on the sidewalk. If you dump solvents on the sidewalk, you go to jail. But if you put it in the fume hood and evaporate it, it's the same thing. So ultimately, I convinced a majority of people to do the right thing. Fire marshals took my side, decided to enforce it. EPA took my side, decided to adopt it and enforce it. The first thing EPA did, they bought EcoFunnel for all their labs. Within a few years, we knew we were going to be profitable. And basically, being who I am, I'm always thinking about the next thing. So I was thinking about putting a product on the market called hypochlorous acid. chloramine for infection. So I started building another company called NovaBay Pharmaceutical, and I incubated it within my CP Lab Safety for several months, maybe close to a year. And then ultimately I created the company. And then in roughly year 2000, I was completely focused on NovaBay, and my wife came and took over CP Lab Safety, and she grew the company. And I left the company altogether. I focused on NovaBay, started raising money. The initial funding for NovaBay came from CP Lab Safety. So we were developing a product for infection of diabetic ulcer, venous ulcer, pressure ulcer. Ultimately, the product is on the market. It's called Avenova Nutrophase, the leading product for blepharitis infection of the eyelid. So I developed those things, did a lot of partnerships at NovaBay with Alcon, Novartis, Galderma, Virbac, China, Pioneer Pharma, Pung Pharmaceutical in Korea. I was on the plane with my business development guy all the time.
I imagine that must have been quite a stressful period, quite an exciting at the same time.
Exactly. Attending lots of bio conferences, attending lots of bio Europe conferences, and also attended Arab Health in Dubai. So a lot of partnerships came from those conferences. But we had a very extensive partnership with Alcon, Novartis, Galderma. When Novartis bought Alcon, they basically, within a year or so, they wanted to cancel the contract we had. We had a very extensive contract. And upon the cancellation of that contract, I had to let people go. And at the time, we had roughly 60 people at NovaBay. I had to let probably a dozen people go, which was a very painful thing to do.
Oh, that's a difficult thing to do.
But on the spot, I said, we have a lot of equipment. We have a lot of analytical equipment. We had an NMR, nuclear magnetic resonance spectrometer. We had mass spectrometry and all that. And of course, we had our microbiology. I said to the people that I was going to lay off, I said, would you guys like to stay on, and I will put a CRO around you? And at the time, we were in Emeryville, and I said, we can call it Emery Pharma or Emeryville Pharmaceutical Services. And out of a dozen people, several of them took my package and left. But several of them, they said, no, we don't want to leave. We'll stay and be involved with both NovaBay and the CRO. So essentially, Emery Pharma was born in 2011.
That's an exciting story. I mean, what a fascinating journey you've been on.
It was sort of, we turned a death into life at that time. Essentially, upon that notice, we essentially formed a new company. And I remember going to Emeryville, California City Hall and filing for the name and registering the company. And that was a nice time. In 2015, we had some clinical failure at NovaBay. Also, we had some success with Avenova. Actually, we were generating lots of revenue, in the millions of dollars in revenue. And my board had changed, and we had some of my big supporters had retired on the board. We had new board members, and there were disagreements on the board. One of the board members, who is an accountant, I won't mention his name since this is going to be made public. Everybody probably will know who that is. Basically, he spearheaded essentially a change in strategy. He wanted to get rid of all the people with lab coat. He wanted to get rid of Emery Pharma as the subsidiary of NovaBay, and I was in disagreement. Basically, the battle of strategy, I lost in that battle. Part of that loss, I also had to throw in my resignation. And the thinking was, upon my resignation, an announcement of downsizing of the company. And we were public, by the way. I took the company public, NovaBay, in 2008. 2007, we took the company public. In 2008, I opened the stock market, American Stock Exchange. It's on my personal YouTube channel. The video of opening of the stock market. Also another big memorable event.
Wow, that's amazing. Very fascinated.
But let me just wrap up. So basically, on the day of the announcement, instead of expecting the stock will go up, the stock fell. Because biotech is essentially rooted in hopes and dreams, and that was not helpful. So I left in November of 2015. I left NovaBay, and I took over Emery Pharma and embarked on growing Emery Pharma, moved Emery Pharma to Alameda, California, where we are today, 20-person, 20 scientists strong. We do what no other CRO does, and I can get into that a little bit later if you want.
Absolutely. We will get into that a bit later. And also, I would like to ask a few follow-up questions about your entrepreneurial journey because you have a lot to share, but we will do that at a later part of the interview. So first of all, I would dive a bit further into the science. And you mentioned that CMC had 3 letters. It has chemistry, manufacturing, and control. So let's talk about the control. And something that has been part of your career for many, many years is impurity testing. So before telling us the story that made you, shall I say, famous, tell us first about what are the main impurities a scientist has to look out for in bioprocessing in particular. Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode.
Excellent question. This is an area near and dear to my heart. For example, in DNA synthesis, during my journey at Applied Biosystem, because we're synthesizing DNA and we're making primers, any bifunctional impurities that would get into the action of the DNA synthesis would just multiply. So those impurities need to be taken out. Not even 0.1% is acceptable. So that's for DNA synthesis. Or any polymerization. You have to care about impurities, especially bifunctional, that could get involved in the polymerization. Most recent experience that I have with nitrosamine is this was a huge newsworthy sort of an event where actually it landed me on national TV on January 9th, 2020. I was on CBS News national TV alongside the head of FDA. How that came about is we were contacted by a pharmacy out of Connecticut called Valley Shore Pharmacy, and they were testing Zantac, which is ranitidine, the active ingredients, and they were testing it by gas chromatography. And gas chromatography, you're heating the sample enormously in order to volatilize certain impurities. And they were observing lots and lots of nitrosamine, specifically N-nitrosodimethylamine, NDMA. Millions of nanograms of nitrosamine, which is just completely unacceptable. NDMA, according to the FDA, you can have only 96 nanogram per day. You can consume 96 nanogram per day if you're taking a medication a lifetime. So they assume if you take it for 30, 40 years, your chance of getting cancer is extremely low. But having millions of nanograms in a pill was just unbelievable. At the time, Valashore and the pharmacy got excited and announced this in a big way in a press release, and a lot of the manufacturers pulled their products, their Zantac, off the market, voluntarily pulled it off the market. We didn't want to have anything to do with that announcement. In fact, he asked us if we would want to be part of that press release. We declined, and because we said we need to do more investigation, we weren't sure about the result. We felt that was due to excessive heating of the ranitidine. And we started our own internal investigation to see what's going on. Our investigation effectively unraveled what's happening with the ranitidine. Effectively, ranitidine was breaking down, and the breakdown product of ranitidine was the NDMA. And on December 10th, FDA press released that if the NDMA impurity is less than 96 nanogram, you can put it back on the market. It was telling in a press release to manufacturers. At that time, we knew that FDA was wrong. So we pointed out to the FDA that ranitidine is not an impurity. It's a breakdown product from ranitidine, which was very devastating, obviously, for the manufacturers of ranitidine. But ultimately, I was invited to be on TV, and the head of FDA was there, and the head of FDA said, oh, we need to do more investigation and all of that. We're not sure about Emery Pharma's results. We need to do our own investigation. Sure enough, they did their own investigation. 3 months later, they came back and they said, everything Emery Pharma said was correct, and in a letter to us, confirmed everything. So as a result of that, we actually were involved in not only the root cause analysis of the nitrosamine in ranitidine, we also got involved in doing root cause analysis, risk analysis for valsartan, which is a blood pressure medication. As a result, I became an expert in all the synthetic methodology that goes on with valsartan and ranitidine and others. So we are effectively one of the leading experts in nitrosamine. And we've been invited to speak at Genentech, Gilead. We were last 6 months or so. Exelixis and others. So we have developed a significant expertise. So in terms of genotoxic impurity, there's impurities like DNA problem where it causes problem with DNA. But here you're talking about a genotoxic impurity, which means this causes genetic toxicity in your gene. It could potentially cause cancer. NDMA causes cancer in every animal they have tested it in. So definitely we want to keep it out of pharmaceuticals, drugs. We want to make sure there's good controls around it. So we advise companies. We're now working for a dozen companies that are putting drugs on the market. Basically, I don't want to scare your audience, but if you're planning to put a drug on the market and you've never heard of a nitrosamine risk analysis, now you're hearing it.
I have a question about that, Ron, because I'm sure some of the listeners are wondering, okay, where is nitrosamine coming from? Because I'm developing a novel process, a new process. What do I need to watch out for? Is this coming from certain raw materials, or where does it come from?
Very valid question. So if you have a secondary, tertiary, or quaternary amine, and if you have some oxidative process, specifically with chloroamine or some other oxidative process, you could form nitrosamine. If you have sodium nitrite, the enemy is sodium nitrite. You could find sodium nitrite in low levels in microcrystalline cellulose, which is used to make the pills. Almost everybody using some form of microcrystalline cellulose to make their pills. It's really a reaction between NO plus nitrosonium ion and a secondary or tertiary or quaternary amine. And if anybody is interested in learning more, they should go to our website. We have a lot of material there. And also, I gave a very extensive YouTube video on nitrosamine based. But of course, I'd love to hear from your audience and see how we could actually potentially help them.
So this means that it's a much bigger risk in traditional pharma processes, and I'd say less of a risk in a biological process. Is that correct?
It's less of a risk in biologic processes, but if you have a small molecule, secondary, tertiary, quaternary ammonium in your process somehow, you need to watch. Their FDA reviewers have seen it all. They're very sensitive about it. As of August of 2025, every manufacturer who has product on the market, who is developing a product, has to submit a very thorough risk analysis, evaluating their incoming material, evaluating their chemistry, evaluating their manufacturing to make sure there is no nitrosamine getting into the act. We had a very bad experience. One of our clients did a superficial risk analysis. We advised against it, and we basically said it's worth doing some testing to make sure that nitrosamines are not there. They spent $6 million making their manufacturing 3 batches, and then FDA basically at the last point, they said, do some testing. When they tested it, they had NDMA 11,000 nanogram for a pill that was supposed to be 40 milligram. It's better to do the risk analysis. So we do a superficial risk analysis at no charge for many of our clients. If they share with us their process, we go under an NDA, we'll take a look, and we tell them how much risk they're going to be taking by moving forward with a very superficial risk. Now, if you have a biologics, nitrosation of a biologics much less of a concern, because basically a nitrosation of a biologic, the CYP enzyme has to convert that nitroso compound into a genotoxic substance, and CYP enzyme cannot accommodate a big biologic, so small molecules are much more prone.
And I would add to that, Ron, it's a good practice anyways to do an impurity risk analysis. This is what health authorities ask for. So it's not only for nitrosamine, but there's all kinds of different impurities. Just not one to mention is also leachable and extractables, for instance, for single-use equipment.
Absolutely. I mean, those things get into the act and they get extracted, and a lot of plasticizers are within these single-use plastics, stabilizers, plasticizers. These are not good molecules to be exposed to. So you want to have controls.
Ron Najafi's journey from bench chemist to serial founder is a compelling reminder of what's possible when scientific depth meets entrepreneurial drive. In part 2, we go deeper into the practical side. We'll look at CRO partnerships, the future of pharmaceutical analytics, and the lessons 3 decades of building companies leave behind. If this episode brought you value, please leave a review on Apple Podcasts or your preferred platform. Thank you so much for tuning in today, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.